So I work on photoreceptors, which means proteins that are responsive to light. And more specifically, I worked on optogenetic proteins. So proteins that when the lights are on, turn on gene activity, and then when lights are off, turn off gene activity. And so my particular protein is responsive to blue light, which does not penetrate very deeply into the skin. And so I work on tools to make them more useful for in vivo applications by changing them from being blue light responsive to developing tools that allow them to be red light responsive, which penetrates much more deeply into skin. And so for this, I rely really heavily upon the photonics core facility on the second floor, and I'm able to use the laser there to help me investigate these sorts of tools that will allow me to have redder wavelengths of light responsivity for the protein. And so other people in my lab also work on photoreceptors, and they use other core facilities in the building, so such as the mass spectroscopy core and the NMR core, to figure out the structures of these light responsive proteins so we can see how they work and maybe see if we can use them in other applications later on.